So very good evening. Very good evening, everyone. I hope there is no echo now. We'll get started in 1 more minute, please. We'll just wait, for others to join. Thank you so much. I hope my voice is audible. Yeah. Thank you.
Yes, Prash. Yes.
So very good evening, all of you. So this is Prash again, organizing this sciversation. In fact, I'm absolutely elated today to have Dr. Ramesh Katam, who is a professor at Florida Agriculture and Mechanical University in Tallahassee, in Florida, in the United States of America. Ramesh, in fact, I'm so happy. I don't know how to express my feelings. I had my first interaction with him in 2007 when I was doing my PhD. And by then, I believe he was into his postdoc and he has already joined his assistant professorship. And we went on to publish the very first peanut leaf proteome. I was a part of a small analysis, a small 1 page or maybe half a page analysis. And that particular paper is one of the reckoners of an analysis of peanut interactions. To just give a kind of an introduction, he obtained his PhD from Mississippi State University. And then he also worked in late 1990s as a research scientist at CREDA, Hyderabad, Central Research Institute for Dryland Agriculture. He also worked for MICO before moving to US, where he obtained his PhD, and then subsequent years of postdoc at Florida University. So it's been almost 2 decades, 19 years ever since he has been working for FAMU, Florida Agriculture and Mechanical University. And his main focus and interests have been into abiotic stress, although he is a so-called plant physiologist. He has a very upper hand in proteomics as well, where he has worked on relative top-down proteomic approaches of various plants such as muscadine, peanut, chickpea, mustard, and also pistachio and other things. He has wonderful collaborations and collaborators in Japan, Iran, and also in some other countries, including India, where he has extensively collaborated with various fields. So he is with his PhD fellows, and his research group is largely working on abiotic stress. So thank you so much, sir, for obliging to my kind request. It's been several days. I've been looking forward for this wonderful session. So we start with your presentation, please. Thank you.
So yeah, good afternoon, good evening. This is morning here at 9:30 a.m. And So thank you, Prashant. This is a nice group, and I really appreciate Prashant doing these kind of things, which benefit younger faculty or younger generation. So Prashant already mentioned lots of things about my education career, so I would not spend a lot of time on that. Although I made some slides uh, because of the time I got in 15 minutes. Let me share my screen. How do I? Okay, I'm going to share the screen, Prashant. Do I say present now, right? Entire screen. Okay, can you help me on this, Prashant? I'm not technical person. So here, my presentation, what do I do? Can you hear me? I can't hear you.
Yeah, sir. Sir, you have to share entire screen
Yeah, I did that, entire screen. And then
and click that and click OK.
it's not letting me do that. So
Uh, I just did that, sir. I don't know why you're not able to see. For example, I just did that. Sir, can you otherwise refresh it? Which browser you are using, sir?
Google, Chrome.
Yeah, I don't know. It should work, sir.
Yeah,
Can
give
you
me a
can you probably just refresh the browser and try again? Sometimes it works.
okay.
Yeah, and now please try, sir. Yeah. Yeah, now we could see, sir. Yeah. Now you can make it full screen. Yeah. Yes, sir. Yeah, it's absolutely very clear. Thank you. Yeah. Sir, you're muted. Sir, you're muted. Ramesh, sir, you're muted. No, here on the mic at the extreme left, mic. Yeah. Yeah. And you can make it full screen, sir. You can make it full screen and share your yeah slideshow. Yeah.
Okay. I apologize for this because I'm not familiar with Google Chrome. I usually do in Zoom, but that's very simple. So did I lose you, or I'm not sure what I'm doing?
Yeah.
So yeah, so this is my place where I am in Tallahassee, and Florida. People may be getting confused with a lot of universities here. So every university is named with some kind of Florida, either in the beginning or at the end of the title. So, for example, Florida State University, Florida A&M University, University of Florida, University of South Florida, University of North Florida. So, like that, it is confusing. So specifically, I am from Tallahassee area, which is north of Florida, extremely. You see the borders and all, Georgia, Mississippi, and Alabama. So that is where I am currently working as an associate professor. And to go with my career, I just want to split my career into three phases, which is phase one is very preliminary, elementary, and where I did high school. Until high school, I traveled across both states, Karnataka and Andhra Pradesh. And then my undergraduate studies were with SV University, Tirupati. And then I moved on to, for post graduation, it's Madhya Pradesh, earlier Madhya Pradesh called Chhattisgarh now, Guru Gashidas University, Bilaspur. So um, I will I will spare this slide at the end for discussion. So why did we, how do we get opportunities to go places, or choose the careers, or what is the transition, what are the difficulties in deciding which career you want to go. We'll talk about that at the end. Um, so here in the second phase, I did my PhD. That is my big turning point. Uh, PhD at MS University, Baroda, Gujarat, and where my research experience was on plant tissue culture. That's where I learned a lot of techniques, plant cell cultures and plant hormones, plant signals, how they behave. So that was a great experience. I spent almost 6 to 7 years. That experience, I had received a research associate position at CREDA, Central Institute of Untitled Agriculture. And there I was doing a peanut tissue culture and cell culture. So peanut is a groundnut. So then later on, I tried to move on to companies where I want to really show and work and do in private companies, private sector. But it was not a very smooth journey there. And so I was doing all crops for tissue culture, genetic transformation. But later on, that is my phase three. So phase starts in my in USA, where I started my career. Before 2004, for 2 years, I worked in Mississippi State as a research assistant, just to get a foot with any position, because I transitioned from India to USA. So which means I need some kind of financial support. So for that 2 years, I was working as a research assistant. And at that time, I got an opportunity to do some coursework. So later on 2004 onwards, I did postdoctoral position until 10, and 2010 to 18 as assistant professor, and 2018 I got tenured, and that will last until 2024 for next another 1 year or two, and then from 24 I'll become a professor. So it is very optimistic view. I do not know what will happen in next 3 years, but this is my prediction and my goal. So uh, right now, who am I and what are my interests? So this is my expertise. I won't say I'm completely expert in all these areas, but I'm mostly focusing on proteomics, and I know a little bit about genomics to understand the proteome functions. And then I take help of collaborators, and I collaborate with people for some bioinformatic analysis. For example, Prashant is one of my good collaborators, and I have some collaborators in Japan. So during when I was doing my research assistant, I was job. I got a PhD at that time from Mississippi State, which is specialized in molecular biology. There, I used to work on stress proteome and genomic analysis of a grape for drought stress. So therefore, I am completely involved in stress biology research, and therefore I got my expertise along those lines from the beginning of my career at in USA. So currently, my major research interests are to understand the cross tolerance mechanism in plants, because plants being sessile, they are subjected to lots of stresses. Then we never know. Sometimes it is salinity, sometimes it's a drought, sometimes it is a nutrition deficiency, and so on. So continuously, plants undergo all these things. So how the plant is capable of surviving? Some plants survive, some plants do not. So that is tolerant and susceptible. We call them in terms of crops, if you want to make a crop research. But here in general, plants, they have kind of stress memory. So that's where I want to really target what kind of stress memory is there. And then when there is a stress memory, can we take the seed and then do a transgenerational research to find out if this stress memory is carried out to the next generations? So it's a good interest in those areas. And also I wanted to, right now I'm working on metabolomics to see how this cross tolerance mechanism in plants is working. And I do have some interest, but I'm not into this. In future, I would be doing for single cell omics, because single cells in different tissues, we see lots of variation. A protein present in chloroplast and same protein present in mitochondria will have slight different functions. Therefore, it's always a good approach to look at the layers of these different organelles, and also the cellular level, understanding the transcriptomics, metabolomics, and so on. So currently my research capabilities. So Prashant said I have full-fledged mass spectrometry. That is not, but I collaborate. So with FSU, Florida State University, which is next door, about 2 miles. So where we have an expert in the mass spectrometry, and there I submit my samples, I work with them, and take the data, understand what is the data is telling me, and how this interpretation can be made, and what is my question, biological question, and what kind of answers I'm getting, and what kind of hypothesis I can make. So it's a kind of generating a model based on these resources and collaborations. So I do collaborate with the University of Florida, which is Gainesville, that is about 2 hours drive, and also I used to have a collaboration with Japan and Australia, but because of the COVID, things didn't go well. And also, this is at Tifton, Georgia. We have a USDA facility where I can grow lots of peanut at their place. So everything is accessible nearby, and so currently, I am trying to develop some omics facility in my lab itself. So there is still a long way to go, and we still have kind of funding, NSF, USDA grants, and so this is what I am currently. And next, so Prashant mentioned that this is our first paper. So when I was doing a postdoc, my interest goes to understand the proteomics. That is where I learned proteomics. So everything I learned here after my PhD. So that is from 2004 until now. That is where I got to expertise in the proteomics. At the time, I used to run one-dimensional gel electrophoresis and used to be happy with all the bands and other things. But at the same time, two-dimensional electrophoresis was just very popular, and that will give a good map of complete proteome. I would say complete. At that time it was complete. So it was some 10, 15 years back. So therefore, I started working on this peanut and two-dimensional electrophoresis, which is a very hard thing. It used to be sometimes we don't get replications. Normally, reproducibility is an issue, and so on. However, with all these efforts, we tried to generate a proteome relief map. That was the first of its in peanut, and then we studied. So in this process, I got interacted with Prashant. So this is a kind of network, small world, right? So where we see he's a skill that he had got good skills in bioinformatics analysis, and he tells me a lot of things about proteoforms and isoforms and so on. So with that help and others, so we developed a paper. It is published in 2010. And see if this is a beautiful picture where Prashant has developed with interactome in orthologs of in Arabidopsis. So this is our first paper, and we didn't stop there. And later on, we tried to see what is available in compared to studies of this kind of proteome, whether it is same in all cultivars, all peanut cultivars. For example, drought. Drought is a major issue here. So in that case, we studied two different plants, drought tolerant and susceptible. And interestingly, we found in a drought tolerant plant, when it's subjected to drought stress or water stress, we found some signaling molecules or transcription factors related. These are induced, either induced or overexpressed in tolerant varieties. So that was a good observation. We published again in 2016, so in about 5 to 6 years later. It took a while to understand these things. And later on, at the same time, I was also doing some soybean proteomics. So interestingly, in soybean, we got a material. So where the breeder has grown this two types of plant material to water stress and also heat. So drought and water stress. So heat is one factor and drought is another factor here. And then he combined both and grew the plants. So then he suggested to look at the proteome profile. Interestingly, when we have that profile, we see that a cross tolerance is shown here. So what is a cross tolerance here again? The proteins that are present in one or more stress and whether that is present in both combined stress. So that will be a memory where the plant carries. So for example, we saw heat responsive proteins and photosynthesis metabolism. Obviously photosynthesis because this is a leaf sample, and oxidation reduction proteins. We found it. And so then we also see mostly the metabolism and photosynthesis are the main functions that are upregulated in one variety, that is a tolerant variety. So that is where we concluded that there may be some kind of phenomena of cross tolerance observed in these plants. And Prashant found some of the one protein, which is a mediator of RNA polymerase transcription, that has a partner, interacting partners in Arabidopsis. So this leads that this kind of protein interactions may be existing in the soybean. So with that idea, again, I went back to peanut. And so this is my graphical cartoon. So where, for example, biotic, abiotic stresses induce osmotic and ionic imbalance. So plants try to do homeostasis in terms of protein homeostasis or genetic homeostasis. They may alter some kind of transcriptions, RNA transcripts. So these are all things occur, and when this could be occurring in either in chloroplast, mitochondria, or in the cell membrane, and then that goes to even chromatin level, where there is a chromatin modifications can also occur, and that reflects on metabolism. So in the end, there could be lots of changes with this signaling pathways. There will be so many post-translational modifications and and changes in the factors. So these are the factors that are very important to find out what kind of factors are triggering, and if these factors are by any means serve as a memory for the plant for next generation. So therefore, what we did was, as we see here, peanut plant has another issue where during the drought, the soil, when they grow in the soil, the drought, the aspergillus fungus is always it's called aflatoxin causing fungus. It's always there in the soil. So when there is a drought, aflatoxin causing fungi, they grow very fast, and they cause multiple. That means we got multiple stress, drought and aspergillus. So it's a good candidate for studying the two stresses. Therefore, we did a control only aspergillus and only drought, and drought plus aspergillus, and then after recovery. So we don't kill the plant until the death, but so we recover the plant and see when we recover, what kind of molecules, proteins or metabolites will come back. So whether it is coming back in both type of plants, drought tolerant and susceptible, or it is coming intolerant. And again, I'm looking at all the tissues, leaf, root, and seed, and how they are coordinating and how they are synchronizing. And then this is all ongoing studies right now. And in next, we will also plan to get the seed from these plants and see if there is any kind of memory that is carried out by looking at the molecular signatures, including metabolites or proteins or transcriptions. So apart from peanut, I did some research with in the same lab where I was doing postdoc on grape, muscadine grape. Probably you may not be familiar with muscadine grape. I don't think it is available in India. But these are the very big, it looks like a small kind of lemon size. So they are hard, thin, the skin is hard, but they have lots of nutraceutical value. So it grows only in Southeast Asia, Southeast US. So which means mostly Florida and a little bit of Georgia and so. And so we studied and we found out some kind of important metabolites, and that while ripening and what stage these metabolites are more or less. We studied certain developmental proteomics here using a different method here. So at that time, iTrack labeling is also one of the tool to understand the proteomics. So 2D from one-dimensional to two-dimensional to iTrack, and then we finally went to LCMS, so liquid chromatography mass spectrometry. So this is about my proteomics research, and as I mentioned earlier, I do have some collaborations. For example, in Japan, one of the collaborator with that, he used to do some modeling, mathematical modeling, and we found out that there is always a perturbation of genetic regularity system while the progression of the climatic change. So we observed some climatic changes in the past, and we see how the genetics of these genetic pathways or in the plant system is changing. So it is a kind of disturbing the genetic regulatory system while the progression of this climatic change. So it is not a good sign. So another collaboration with West Virginia, and one of the student, she did some work. We have been collaborating under the USDA project. So we found out that there is a chromatin modifications and transcription regulations in watermelon when there is a gene duplication from tetraploid, diploid to tetraploid. So watermelon is a tetraploid. So this work was published in Plant Journal, and this was not so very nice work that they actually put their cover page. So currently the student is in Max Planck Institute, so which we're proud of that. And well, so just don't worry about these numbers, but so far without money nothing can be achieved. But so I could at least get some money up to 2 million and little more than 2 million from NSF, USDA, and NIH. And so these are some of the things that I achieved so far. Right from the beginning career, I got fellowships from Japan Science Society for Promotion of Science. I would suggest encourage younger generation to consider this because it is straightforward, and you find a mentor there and a collaborator and work for that. Prashant will tell us more. And also later on, as a routine, so as you do good, so Family Emerging Research Award, Teacher of the Year, Advanced Teacher of the Year, and a couple of travel grants to travel overseas to France and Canada to attend and present my research there. So I traveled even to Australia, collaborated with in Macquarie University, where I did some work on grape. So yeah, and other than that, current activity, I do have a summer research grant, which NSF funded for last 12 years. And it is currently still going on for next 3 years. So under this grant, students, I pick students from all over the country. Every year to 8 students, they come to my lab and they learn the techniques and skills, and they try to publish so far. And we invite guests. So this person is from Australia. She is a proteomics person from CSIRO. She's talking to us. So like that, this is my activity in the summer, and regularly otherwise teaching is my major area, and I do make research in the collaborative basis. So I have some facilities, and still I'm developing some omics facility in my lab. So here I just wanted to share this with you. We talk lots of research and say a lot of things, but how much research is reproducible? So that is a big question. So the Nature, I got it from Nature. So in 2013, Nature has asked all the authors so far to present the reproducibility. According to the survey, they found out 86% of the respondents considered poor reproducibility. So that means only 14% is reproducible. That is a very challenging stuff. So think about it, and how to solve this problem. I have some suggestions here. So in order to achieve a reproducibility here, so have a transparency in your research. So try to document yourself and try to do multiple times replications and replications. Biological samples, 3 replications, it's not good enough. All the papers take it. I would go 6 replications and more. And then, so you are not alone, you can't do alone. So you need to develop some collaboration across the world, across the planet. So anybody, so just email them, find them in, nowadays you got all the resources, but in 10 years back, it was not so much. You got Twitter, you got Facebook, you got LinkedIn and all stuff. And diversity is very important. Get in diversity. So talk to people across the East, West, everywhere. So somebody may not be interested, but there will be somebody looking for the collaborations. And complete literature search is very important. Without doing literature search, you don't know what exactly we are going to be. If it's already published, we don't know, and then we ignore, and we don't cite them. That is not right way. And Even if you find a literature, check for authenticity, that that is a paper is worth spending time on reading it or not. So not just based on impact factor, but impact factor is always kind of misleading. So I don't go by that, but always try to see. For example, when I said in the last few years, nature itself, 86% is not reproducible. So with this few comments, I would rest to Prashant. And then one slide I just missed because I will spend for my discussion. Thank you for listening to me.
Yeah, yeah. Thank you very much, sir. For a scintillating presentation. That was certainly a kind of an eye opener. I mean, um, I echo the sentiments that the reproducibility is very, very important, and the current generation should really take this forward. And I've really learned a lot of things from you ever since I've known you since 2008. In fact, you'll be surprised to know that, dear friends, the very first interaction that I had was in 2008 February. And by the way, we never met, sir and me. We never met. We missed on occasions, couple of occasions. Somehow we couldn't meet. But 2008 February, or even before that, 2007 October, I believe. That was how we came with interactions. Our first paper, in fact, my major first paper was only published in 2010 with him. So, which means it took almost 2 years, 2.5 years, working with him. Sir would ask, several questions left and right. And I was also in my early postdoc phase at that point of time. I learned a lot of things, and that's what matters for a kind of a reproducibility research. So moving forward, I have a lot of questions to sir. The very first thing, I would gratefully acknowledge that I'm also a Guru Ghasidas University student, sir. I finished my MSc in 2002, and so sir is my super senior, and that was how we do it. And I really take this wonderful thing here. Sir, the very first question is what made you to focus on stress biology? What was your inherent interest? Was it your academics, or was it your MSc times, or was it your agrarian background? Could you please enlighten us?
Yeah, so it's not something very. I have no interest in to study this or that. But when I joined and started working in plants, so obviously the plants do have continuous stress. It's not that like humans, they go and take a shelter and they'll be survived. But plants are not like that. So every time plants do have stress one way or the other. So therefore, it is very important that how these plants are doing a lot of good things when there is so much of climate changes, day-to-day changes, not even forget about year changes, but every day it changes to things. So therefore, I started working on stress. I prefer abiotic stress because it is easy to handle. It doesn't require any inoculations and all, growing of the fungal cultures and the bacterial cultures. It's a kind of another additional stuff that we should take care. But abiotic stress is very straightforward. We don't have to worry. But still, combining the biotic and abiotic is very interesting because the biotic, like microorganisms, always interact with the environment, and that environment will create another set of environment to the plant system, ecosystem. So it's very interesting. So that's where
Right.
I wanted to.
Wonderful, sir. I mean, so at the helm of this wonderful experience that you garnered in India, sir, working for MS University of Baroda and MICO and CREDA, you had a major transition to US. How good was, or how tough or how challenging was that transition, sir? All the way, from India to US. How did you cope up the acclimatability or acclimatization and all those kind of things?
Yeah, that is a very great change. In fact, I never wanted to go and serve a different country. I am so much of patriotic, so that is true. In fact, that's why I decided to stay and work for and be a good, make great things in the private sector, because I see that government sectors back then, 20 years back, it was not so bright, not so encouraging. So it's a very difficult situation to survive under the sector like government sector. But therefore, I thought, okay, private sectors are very great, and I can make a lot myself comfortable. Unfortunately, things are not that great. Over the time, the private sectors also they behave crazy. So that is one of the frustrations we feel. So it's not just me, like me, there are so many people. So they keep changing jobs from one company to other company. I have my own colleagues, they switch companies every 3 4 years. So it's not kind of stuff that I want to. The finally I said, when instead of changing all these companies and I'm not making any nickel and dime, so one big leap, one first step, let it be whatever it is. So I started working as a research assistant, which is not even any kind of, just I can't pay my own rent for a couple of years. So that was the stage I went started, and I know it's going to be good. The good days will come ahead. So that was my very great step, which I surprised myself. So after that, I didn't look back and keep going and going.
Yeah, wonderful, wonderful, sir. So moving further, I mean, I know amongst your collaborators, Dr. Sakata from Japan is among the topmost, I would say, collaborators, and we just came to know that you were telling us he recently passed away. I had an opportunity to meet Sakata in Singapore, I believe, way back in 2009 or 2008, and I mean he was an amazing scientist. So could you please enlighten your relationship, collaboration between you and Sakata, sir? Yeah, please.
Oh, I can't forget that. So he's such a great personality, great man. He loves me, I love him. So that is what kind of. So we met after meeting first time in 2010 as a JSPS fellow. Uh, so we don't know each other, but again we were discussing in terms just like you. I was talking to him in the past about the paper collaboration. So which is what generated that JP paper, Journal of Proteomics, for peanut drought and drought susceptible plant. So he's a great, and he is a very very simple person. That's what I learned a lot of things from him. That's why I encourage diversity. If you look at what the other persons are, how they are, forget about all. I mean, ignoring the history. So we have a very bad history uh, during the world wars and in the past also. So we don't want to consider those things. But moving ahead, the simplicity and the diversity is very very plays very major role. It changes your way of thinking, and it helps in our research as well. So Sakata is a great guy. So I met 4 times. I went to Japan 4 times because of him.
Wonderful, sir. I mean, that's indeed pressing. I'm sure this would be a fitting tribute to him. So moving further, ever since you started working on stress biology, a lot of things has really happened in plant sciences, right? From the good old, the microarray technology to the next generation sequencing, and even in the proteomics, you have been working on various things. Several mass spectrometry instruments, right from the LCMS, GCMS, the MALDI-TOF, the CELDI-TOF, the Q-TOP, and all those kind of wonderful instruments have really popped up. And yet for some reasons plant biology remained or still remains at least 5 years behind what an animal researchers probably they would be doing. Do you agree to this particular context, sir, in terms of the technologies or in terms of the pervasive technologies that are coming up, and also in the context of how the scientists and scientists with plant biology grow?
Absolutely. So it's a very important question actually. We looked at metabolomics, proteomics, or genomics for that matter. Um, Genomics is pretty close to the animals sciences in plant sciences as well. But in terms of molecules, proteins, and metabolites, there is a very little progress in the past, I would say 5 years. There is no way, there is nothing changed in last 5 years between now and then about metabolomics because of the complexity nature of the plants. Because today you see a metabolite and tomorrow you may not see the same metabolite in the plant. So much of interaction, and therefore it is very important that the there is a chemo environment, chemical environment in the plant changes every season, and you do not know whether the same things will appear in the next year following the same conditions. So there is so much of interactions like bacteria, fungi, they go interact with the plant like herbivores or the insect. They feed upon the plant and they damage, and plants will produce some kind of glucose analytes chemicals. So this is a constant, continuous process occurring in the plant kingdom. So therefore, it is very difficult, and also we don't have any markers for this definite metabolite markers, chemical markers, as you see in animals or humans. And again, look at the post-translational modifications for proteins. You do not know what is going to happen when the gene is translated to a protein, whether this protein is going to make it or it is going to be modified for different kinds of stresses. So for one type of stress, the modification may be different. Another kind of stress, there won't be different modifications. And also, again, at the organelle level. So the protein present in a nucleus, the same protein present in cytoplasm, have a different function altogether. So therefore, it is important. That's what the technology is advancing towards single cell metabolomics, single cell omics, or organelle metabolomics or omics. So hopefully the omics studies and understanding, putting together with the help of bioinformatician. Again, we have so much data. We can generate so much of data with machines, but how do you address the question? How do you answer the question? Who will help us? So again, you.
Yeah, I got it, sir. I mean, wonderfully inserted, sir. Thank you so much. So moving further, one of the intriguing things that has happened, I mean, something moving towards this particular context, as someone who has shown some kind of expertise in NGS, I always feel that the plant exomes or plant exome sequencing was the key at some point of time. But unfortunately, the plant exome sequencing, the very first plant exome sequencing was employed only some 3 years ago. Only they just thought of doing the plant exome sequencing only in 2018, I believe, or 19, I believe. Very 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 sad. State of things. But do you think the reason d'etre or the reason behind such delay, is it just because of the characteristic chromosomal based markers, or the so-called sequence tag sites that are largely used with respect to the nils, the near inbred lines, or recombinant inbred lines, or maybe it is just because that particular stage is not really developing in plants? Why is it the researchers have not really thought of plant exomes? What is your opinion on this?
Yeah, it is a difficult question out of my area, but I'll try to address. Most likely, the plants we are focusing on crop plants, for that matter, at large scale. So if you look at the studies between crop plants and other plants, so you see lots of literature from crop plants, and they are focusing. So with that, putting all these different germplasm, so we are narrowing the genetic diversity here. And again, we are going back to the chromosomal location and all. So people are not really studying in depth about what you just said, the exome sequencing and all. So they may not be ready yet to understand the application of those studies in the plants. So I'm not sure about animals. Do you think animals is the same thing?
No, I don't think so, sir. Animals, for some reasons, maybe just for better quantifiable traits or for better, production traits, just because animals, can be better, taken for granted, for production of milk or probably for production of meat or other things. There was a kind of an upper hand in that particular perspective, and Probably, I would probably look into the other aspect. Maybe animals are more surrounded by the influence of epigenetics, or maybe they are more heralded by the epigenetic influence when compared to the plants. And you find that kind of phenotypic changes more in animals and humans when compared to the plants. I mean,
Actually, plants do have lots of epigenetic modifications. Only thing we never focused on that. That is the thing.
right. I mean, what I meant was there is an epigenetic influence heralded by plants as well, but yeah, maybe that study probably is not really taken up just because you don't see that phenotypic picture or phenotypic correlation as such when
Right,
compared to animals and humans.
right, right.
Uh, Sir, moving further, sir. Over the last 10 years, I believe, ever since the conference on parties have come up, there is a large amount of focus on the climate change, and a lot of emphasis is based on the role of plants, where we need to muster the support of growing a lot of plants, vivid plants all together across different communities. Um, the influence of small number of pesticides and all those kind of things. So, as a plant physiologist, what is your take on this influence of climate change and the role of plants towards taking up the abiotic stress?
Yeah, that is climate change is always there, and it has been changing not just now, but since the evolution, since the formation of Earth. But for our benefit, for humankind, what we need is, say, Project 2050. So the growing population and the projection doesn't match. So, for example, wheat production will not match to 2050 population as the current projection is by FAO and other agencies. Um, so 2050 is not too far, and we need to do a lot. So therefore, so lots of alternative agriculture systems are being developed, and in order to feed this population. And secondly, most of these crops, they go under controlled environments, so like greenhouses. So a lot of greenhouse culture will develop now. It's coming up actually, and of course, who has to pay? Consumer has to pay. We have to pay. The humans, when we are creating so much and growing the population, so we are all equally responsible to give back and pay for them, for these things. So climate changes continuously happens because over the past 10 years, or there is some, I forgot the data, but there is an increase in temperature by 1 degree every year. So that
Yeah.
tells a lot, because plants, they have no place to go unless you protect them by the greenhouse or shelter. That's what we need to do now. Like animals, we have to treat them plants also.
Right, right. I mean, well said, sir. Sir, when you were saying about wheat, one thing, struck my mind. I mean, I worked for about 6 months in P.K. Gupta's lab in Meerut, and I know, wheat is a kind of exploit organism, and working on wheat, that too during my prime phase of my career when I just finished my MSc in 2002 and 3. That was a kind of a very Herculean task, but even now I still uh, find it very uh, I mean what do you call surprising that not many researchers were really successful in making this hexaploid wheat into the realm of understanding better genetics. I know people are really growing a good number of varieties and all, but can't we just take some kind of influence of genetics even in the diploid uh, organisms in plants, just like peanut or any other things, and transfer that particular knowledge in what we call it as in wheat? If that particular study at the proteomic level, and then employing the reverse genetics right from to the gene, if that can be employed, that can really show some kind of wonders. I'm not sure if this has really been taken up so that a better dissemination can take place.
Yeah, I agree with that. But the question again is, how much time we have, and how much we need to spend on that, and are we ready for those kind of techniques. And alternatively, people are looking for alternative crops. So if not wheat, what else we eat? So that's why so many crops have come up, which is very simpler to deal with that, because if you want to put any budget on what you are suggesting, it's very time consuming and also very expensive at the moment. So therefore, um, it is kind of we don't have much time. That's what I would say, because of the situation.
Right, right. No,
So,
well said. Yeah, wonderful. So final couple of questions before we close in. Sir, what is a major difference that you really found in academia in US and in India?
yeah, very, very, very complicated question, but I'll try to see that if I can do justice. Academic situation in US and India. So there's no doubt Indian teachers are great. They teach a lot. They teach good stuff here, and I learned a lot from Indian teachers in my when I was there. Um, but the only problem with the Indian academic system is that there is a big difference between one school and other school. So how many people have approach or capacity to go to only that small number of schools? That is an issue, and therefore, and nowadays this academics have become kind of business. Of course it is a business, here also it is a business, but in here, therefore we in India, not many people are getting same kind of education as it is supposed to be. So sometimes you are on your own without any resources. You are on your own here as well, but we have resources. But there, with no resources, for example, mentoring, we don't have good mentors. There is no mentor kind of concept there. Mentor means it's not definitely a teacher. Mentor could be your younger brother or older brother or your relative or your person whom they can, without impartially guide you what is your potential. So that's what is needed to every person in this education system.
Well said, sir. I mean, I completely agree. Sir, a kind of uh, one important question which I usually ask all my guests in conversations. So, were there any occasions where you try to disagree in an agreeable way with your mentors or with your bosses or with your, maybe with funding agencies or anyone? The reason why I really want everyone, especially the young generation, to learn from this is somehow, we seldom, agree to disagree. We don't try to disagree in an agreeable way. We always, come up with certain fighting conundrums always. So could you please highlight any one example so that the young brethren can learn? Yeah, thank you.
That is a very routinely happening situation. You always disagree. There is always a situation where you will disagree with. So forget about the outside. Within the home, you disagree with your own people many times. But what is the solution is sit together and solve the problems. But in coming to your profession, if you will disagree when your boss is not giving you enough funding to do some work. So at the same time, that mentor, that your boss may have some limitations, which is not able to explain to you properly, and we are supposed to know that what is the limitation there. So it's a kind of developing a transparency. Okay, I got this much of funding money, and I can only do these things. So that is the kind of thing which not many people will share those limitations. People will feel shame, or people will think that if I say my inabilities to this student or this mentee, they will look down. That is not true. The first thing I do to my students whenever I say, this is my limitation, I have problems, and I don't have any great facilities. So you need to work on yourself and resolve and see, let us see how we can come up with. So when you, first of all, you tell them openly about what is your potential. You can't say that I know a lot of things and always you come to me. It's not, nobody knows a lot of things. So if you say I have only my limitation, my knowledge is only this much, I can be only expertise in this much. So that is about it. So kind of ego and issues, kind of false claims are the main drawbacks, creating problems to uh, system in the system.
Wonderful, sir. I mean, very well said. I mean, thank you so much, sir. I think, Abhijit Roy as a question coming to the climate change, what do you think of synthetic biology's importance in crop improvement like carbon fixing efficiency?
Yeah, synthetic biology is a very hot topic, and NSF is funding, pumping lots of money there. So that's very important because we need to engineer the system. We need to, it's kind of biochemical pathways we need to do uh, to sequester this carbon synthesis. And mostly we are so behind in coming up with biofuel crops. So that is another thing which is important to somebody to focus on. Yeah.
Yeah, very well said, sir. Yeah, thank you so much, sir. Abhijit, I hope that answers your question. Sir, before we close it, do you have any questions or any quick comments that you want?
No, it's a great object. You have a good objective in mind. You're doing great. So I hope these kind of things will foster and help students to go in the right direction. But always, one suggestion to my younger generation is that don't do things just for the sake of doing, and do it by heart and do it with commitment. Then results will come automatically. So it is not a kind of philosophical thought, but that is what it is. Because to my experience, I don't know how many thousands of two-dimensional gels I ran. I did. So to get one or two papers with that, it was so tedious. So that is the thing, and repetition, practice, and understanding, serving, looking at the literature, updating your knowledge is very, very important. Today's technique will not be tomorrow. So next for 10 years, your research will be gone. Like 10 years back, we published. Then that is not even now existing. 2D, nobody does 2D, but so what we gave a message is that we got some 300 proteins, but we can get more proteins now. With LCMC, MS technology, we are getting thousands of proteins. So that's what we need to understand. With the limited facilities, what we have techniques, we should try to give a very good interpretation of the data what we have. There is a questions.
So there's one question, sir. Yeah. Anuj asks, will research reproducibility in life science changes with nature? Or in terms of the in vivo or in vitro things, maybe, yeah, that is now more clear. Yeah.
Research reproducibility will change in terms of in vivo and in vitro. In vitro conditions, it's not change, but in vivo it might, because when you grow so in terms of plants again, when you give a condition, the same condition sometimes may not be existing in the same way. Uh,
Yeah, yeah, yeah. Anuj, please unmute your microphone and ask. Maybe that will be more clear, please.
yeah,
Am I
let's go ahead.
audible?
Yeah,
Yeah.
please, yeah.
Yeah. Thank you so much for that enlightening talk. Basically, I am asking that because there is a chance that once you have some kind of result, and in the second chance with the same conditions and same alterations, there might be a chance that you get varied result depending upon all the physiological conditions that are different. Because in life sciences, there is nothing that like a physics and chemistry that we are one or again and again coming on the same point. There might be a slight 0.1 or 0.2% difference. So that's I am asking that
Right.
what sort of, because you are explaining that there is a reproducibility aspect. So I want to know that will this be 0.1 or 0.2% slight changes, or is
Yeah.
applicable, or
Mhm. There is always a statistics applied to. That's why we do some statistics, and always there is a variability. So um, let me compare this to a drug that we are taking. Instead of 0.001 ppm drug anesthesia, if you give 0.005, that is going to kill the person. So similarly, that means it's a no game, right? So therefore we need to really focus on reproducibility, whether it is a life sciences or non-life sciences, because um, Of course, there is a variation, right? We can accept 0.0005% of changes is always there. That's why we quote within that limitation. And if we are not getting, uh, do multiple replications and replications, and then remove the outliers. So between 1 and 10, you will see 1 has a very little number and 10 has a high number. It's only one sample. So delete that number. Don't consider that experiment is done. So take the other 8 samples and do the statistical analysis. And if it works, then so always we need one bioinformatician and one statistician in our life systems all the time. Otherwise, we cannot go. We can't move on. That's what is metabolomics. People don't accept if it is not even 5 replications. That is the.
Yeah, very well said, sir. Yeah, thank you so much, sir. One final question from Alekia Lu. What is the driven force or the motivation when you're failing in an experiment or when you are not getting results? I mean, how do we touch the base? Yeah.
So when you fail, first thing you go and watch a movie. That's it, and then come back next with fresh mind, with a fresh thought. That's all I suggest, because I do the same way. If I am messing up things, I just ignore that lab for a day. I don't go to the lab for a day or so, and then I look at again freshly what could have gone wrong, and I go to my notes and see is there any mistake I did, or did I ask somebody? If you take somebody's buffer solution and use it, we do not know how the buffer was made, and you may make mistakes throughout your experimentation. So you need to go back and what is it called? Double checklist. You need to make a checklist. So when you are doing a PCR, so you add everything. So check. So add 1 check, add 2 check. So then you should be perfect. If not, second time, third time. So recently I had the same problem. No matter how many times I run a gel, the bands are not moving down. So what could be the reason? Logistically, it is a tank buffer. So, but tank buffer we changed. And finally, I asked somebody to do, and they also got the same thing. Then that is my lab problem. So then I asked some other lab to do the same thing. They got it. So I have some problem in my lab. So that is where the basic thing goes back to the elementary level, where we need to like rebooting. Like that's what you do when computer doesn't work. So you need to start. There is a solution for that.
Wonderful, sir. Very well said. And I would like to thank and congratulate the 3 wonderful young scientists here, Abhijit, Tanuj, and Alekya, for asking wonderful questions. Really, I'm really glad after so many days, we have had very good questions and wonderfully answered by Ramesh sir. And I hope you all will agree that we should not wait a minute, for taking a good photograph with Ramesh sir. So all of you, please switch on your camera. And yes. So, sir, we have all wonderful people here. We have Sugu here, who is our Secretary of BioClus. Alekia Lu is a student of Lovely Professional University. She is doing her internship at NIN Hyderabad. Anuj is undergraduate student. Shweta, Hari, Hari and Dhananjay. These are all new faces, in fact, but I'm really glad they are made here. And I could see Suresh here. Yeah, and there were a few others who joined and who left, because of some meetings. Yeah. Thank you so much. So all smiling faces, all of you, please. And whoever gives a big smile, I think you'll get a kind of a chocolate at a later point of time. Okay. Thank you. Yes. 1, 2, and 3. Yes. Yes. Thank you. Thank you so much, sir. Ramesh sir, for your tremendous time and energy. I'm really grateful, for really, giving a kind of an inspiration, today. Thank you so much, sir. Thank you.
Thank you all. Take care.
Thank you so much, sir. Thank you so much, sir.
Thank you, Sunath.
Thank you, sir. Thank you, sir. Thank you very much.
Yeah.
Thank you. Bye.